Welcome to From the Spectrum podcast. This is a podcast about autism. It is my goal to explain what is autism. I plan to use a mixture of scientific literature, personal experience, and opinion. With opinion, I will explain why I feel the way I do and give examples. I will provide links to various references for each episode. For each episode, we will discuss various aspects of autism. For today's episode, it is my pleasure to announce the guest, Dr. Leanna Hernandez. Dr. Leanna Hernandez is an assistant professor in the Department of Psychiatry and Biobehavioral Sciences at UCLA and a member of the Center for Autism Research and Treatment, or CART, the Center of Neurobehavioral Genetics, and the Brain Research Institute. Dr. Hernandez also serves as a co-director for the Genetics, Genomics, and Informatics core for UCLA's Intellectual Developmental Disabilities Research Center. In addition, Dr. Hernandez directs the Hernandez Lab at UCLA, where they examine genetic and neurobiology of autism, schizophrenia, and other related psychiatric disorders. Our discussion includes genetic variations and brain connectivity in autism, oxytocin and oxytocin receptor gene, sex and diagnosis differences in autism and brain connectivity, and the future of autism research. With the sex and diagnosis differences, you have autistic boys, non-autistic boys, autistic girls, and non-autistic girls. So 4 comparison groups. As you hear Dr. Hernandez explain this connectivity, think about autism, what you know about autism. Remember the social communication and interaction challenges and the restricted repetitive behaviors. The preference for routines, sameness, and those preferences for restricted, fixated interest that are perfect in intensity or focus. You know the DSM uses abnormal, but to say abnormal with interest, intensity, or focus is wrong by them. Now, think about how the biology that gives us autism provided those behaviors, those routines, the sameness, and so-called restricted behaviors. They are safe and predictable. The biology that gives us autism creates a chaotic outside world, and this connectivity gives us poor adapting skills. With the poor socialness and restricted behaviors, we have limited adaptive responses. And an unpredictable, chaotic outside world requires adaptive responding. The biology that gives us autism allows us to be comfortable within ourselves. And that can be a path for finding and developing superpowers. And now, my discussion with Dr. Leanna Hernandez. Dr. Hernandez, thank you for joining. Why don't you start by taking us to the moment you realized autism was going to be a part of your professional life, and take us all the way up to the present day.
Sure. So I first started working in the field of autism after graduating from college with a degree in psychology, and I started working as an in-home aide for kids with autism. At that time, I got to know a lot of the different families and the challenges that they were facing, became familiar with many of the different behavioral symptoms associated with autism. and just became really interested in it more generally, and really fell in love with the kids and families that I was working with. After that, I decided that I wanted to learn more about the brain and brain development in general. I've always sort of been interested in development. And so I went on to work as a research assistant at UCLA in a lab that collected MRI scans, so magnetic resonance imaging scans of the brain, during childhood and adolescence, both in typically developing kids and in autistic males and females. And so from there, I then went on to become really interested, obviously, in brain development, and I learned more about the genetics of autism, and went on to do a PhD in neuroscience, also at UCLA, where I studied the effects of different genetic variants on the oxytocin receptor gene and how they're related to brain function and connectivity. After that, I continued to be interested in the genetics of autism and did a postdoc at UCLA, again, really focused on psychiatric genetics, and began to investigate genetic variants associated with autism through genome-wide association studies, which is some of the work that we continue to do today.
Oh, wow. Okay. Tell me about the creation of the Hernandez Lab.
Yeah, it's very exciting. So we just opened up our doors in December of 2022. So I currently lead a small group at UCLA in the Department of Psychiatry in the Semel Institute. And so our work really continues to focus on understanding brain development, not only in autism, but also in other related childhood neurodevelopmental disorders, including psychosis and ADHD. We're also very interested in some behavioral symptoms that sort of cross diagnostic boundaries. So, for example, issues with sleep or sensory processing. And so these are some of the things that we're continuing to investigate. We conduct MRI studies, and we're also interested in identifying the genes that might be associated with some of these different behavioral traits.
Okay. So something that interests me is the downstream effect of that might be autism, or it might be OCD, or it might be schizophrenia, etc. Could you briefly explain maybe what is known about what causes the differences in the disorder?
Sure. Yeah. So a lot of the different neurodevelopmental disorders that you just mentioned are highly polygenic. And so what polygenic means is that there are many genetic variants across the genome that are associated with those different disorders. So, for example, in the case of autism, it's hypothesized that hundreds of genes across the genome are associated with the disorder. Now, the genome-wide association studies that we've performed so far haven't identified all of those genetic variants. The identification of those variants is really a function of the size of your study, the sample size. So what we know is that some of the genetic variants, as you mentioned, that are associated with autism might also be associated with ADHD or schizophrenia. And this would indicate what we might call a positive genetic correlation, meaning that the same genes are influencing two traits. And so, there are some of those variants that will impact multiple traits. And then, of course, there may be others that are specific to a particular neurodevelopmental disorder. And so that area of research is still really ongoing, identifying those shared and unique genetic underpinnings of the different neurodevelopmental disorders.
Okay. It always seems like there's a lot of variability and moving parts funneling down into the individual. Let's start with one of your earlier research papers released in 2017, if you don't mind, in molecular psychiatry. Take us through what questions were asked, or how was it determined. You mentioned earlier, as you're in your education development, you studied oxytocin receptor quite early, and then was just around that time frame, or um, let's just walk through that paper.
Okay, sounds great. Yeah, so this was a paper focusing on genetic variants, common genetic variants on the oxytocin receptor gene. And so what we're looking at here is single nucleotide polymorphisms, or SNPs. And so these are common genetic variants that occur across individuals in the population. They're very common. So by definition, they occur in at least 1% of the population. And they're variations in a single, base. And so you can have, somebody who might have a G genotype, GG, or AA. And so basically we're looking at differences between people who have these different genotypes at a specific genetic variant. And so here we wanted to focus on variants on the oxytocin receptor gene, and we chose that gene for a couple of reasons. First, because there had been animal studies that showed that there are differences in oxytocin receptor gene expression in subcortical brain areas that are involved in reward and social processing. And so some of the really foundational work in this area has been done in animal models, where they found that, for example, in prairie voles, which engage in high levels of parental behavior and are monogamous, if you look at brain slices from prairie voles, what you'll find is really dense staining for the oxytocin receptor in these subcortical reward-related brain regions, including the nucleus accumbens. Now, you can contrast that with, for example, the meadow vole, which lives in solitary burrows and is non-monogamous. And what you'll find is much reduced staining for the oxytocin receptor in that nucleus accumbens brain region in those meadow voles. And so that's, sort of an interesting starting point, some animal work showing that there might be differences in gene expression in this brain area in these two species that are quite different in terms of their social behavior. There had also been some studies suggesting that there might be a link between different genetic variants on the oxytocin receptor gene and autism, so some family-based studies. Although I do want to point out that more recent genome-wide association studies haven't found a link with the oxytocin receptor gene. So a lot of the work that we were really doing, and as you mentioned, this was back in 2017, was more grounded in this, animal work, which had shown an association with social behavior, parental behavior, and bonding. And so what we did there was we looked at 4 different genetic variants on the oxytocin receptor gene, and we made an additive score across those different genetic variants. So how many autism-associated alleles does a person have, and adding them up across those 4 different genetic variants. What we then wanted to do was to look and see how a person's sort of additive genetic liability for autism on the oxytocin receptor gene might be related to brain connectivity. And there we were really interested in, again, connectivity of reward-related brain regions, specifically focusing on the nucleus accumbens. And so I know you've talked in some of your previous podcasts about some human work that has shown that there may be differences in nucleus accumbens activity in autistic people when viewing, smiling faces, different sorts of social rewards. And so that was sort of the basis for this work as well. So what we did is we had about 40 autistic children and non-autistic children come and do an MRI scan. And what we did was we looked at connectivity of the nucleus accumbens with different brain regions. And so how this works is that you would take a seed, a region in the nucleus accumbens, and you extract brain activity from that region. And then you look to see how that brain activity is correlated with every other region of the brain. So are there brain areas that are increasing their activity in synchrony with the nucleus accumbens? And also you can identify brain areas that are sort of anti-correlated, right? Where the nucleus accumbens activity is going up and the activity in these other brain regions is going down.
It's like a seesaw.
Yes.
Envision it. Okay.
Yes. And so what we did was we looked to see, first, what does this connectivity of the nucleus accumbens look like in our autism group and in our other group? And so what we found was that, using this seed in the nucleus accumbens, we found really robust connectivity between this hub of the reward network, the nucleus accumbens, and other subcortical reward-related brain regions, as well as areas of the frontal cortex. And then we also saw some negative connectivity, so anti-correlated activity with subcortical brain regions like the thalamus and also visual cortex.
In autistic groups or the typical
In
group?
general, in both
That's
groups,
right.
in both groups, yes. We actually did look at the differences also between the autistic group and our typical group. And what we found was that the typical group had more connectivity between the nucleus accumbens and an area called the precuneus, which is thought to be really important when sort of thinking about the thoughts of yourself and others. It's the hub of a network that we call the default mode network, which I'm not sure if you've discussed yet already on your podcast, but
Not
maybe
yet, just briefly.
Okay, great. So from there, what we did was we really wanted to see, right, the impact of these oxytocin receptor gene variants on this brain connectivity. So we want to see how is connectivity changing as a function of increased dosage on this particular gene. And so what we found was that in our autism group, having more genetic variants associated with autism on the oxytocin receptor gene was associated with reduced connectivity between the nucleus accumbens and other subcortical brain regions, including the caudate, putamen, and also frontal brain regions in the anterior cingulate.
So the dorsal striatum, the caudate and putamen, for the listener, I've discussed this. That's more of like what I know it as stimulus responses.
Sure, yeah. It's involved sort of in, implicit learning, and you're right, stimulus response, correct.
Okay. I've talked a lot about stimulus responses. So I just wanted to make mention of that.
Yes, perfect. And so what was interesting is that we found a quite different pattern in our other non-autistic group. So in non-autistic individuals, what we found was that having more of these, they still have the autism associated alleles, right? As I said, these are quite common genetic variants. So even in the non-autistic group, what we found was that having more autism associated variants on the oxytocin receptor gene was associated with increased connectivity between the nucleus accumbens and regions in the frontal cortex, and particularly the frontal pole. And so we were sort of curious, what is this difference in connectivity? What does it sort of mean? And so what we did was we extracted the connectivity values between that nucleus accumbens and frontal cortex. And we said, okay, what sort of behavioral measures might this be related to? Mm-hmm. And so we took that activity and we looked at its relationship with scores on the social responsiveness scale, or SRS. And so this is a parent-report measure of autism-associated behavior. It is, adequate for use both in autistic and non-autistic groups.
A very popular instrument too, yes, during the autism assessment.
And so what we found was that in this non-autistic group, this increased connectivity between the nucleus accumbens and frontal brain region was actually related to lower scores on the SRS, indicating more sort of typical behavior, less autism-associated symptoms. And so we took that to mean that that might be sort of a compensatory mechanism, this increased connectivity between the nucleus accumbens and frontal cortex, that, when a person might have more autism-associated variants on the oxytocin receptor gene, the extent to which you can sort of upregulate this connectivity between the nucleus accumbens and frontal cortex might sort of buffer and, result in more typical social behavior.
Yeah. The kind of the prefrontal is my vision of it is, I always say it quiets things so that we can properly evaluate things.
Right. Yeah, that's a big role of the prefrontal cortex, is sort of top-down modulation of some of these subcortical brain structures that we were just talking about.
Mm-hmm. So that's a big finding, that difference. Whenever you first saw the total number of L's, I'm not sure if I got that right, they seemed to me like they were pretty similar. Is that the population has that 1%? Is that from
So you're saying the total number of autism-associated variants between the two groups?
Yes.
Yes, they were very similar. That's right. As I said, these are very common. Everybody has these single nucleotide polymorphisms, these SNPs, and so they were present in both groups.
Okay. So with that SRS, what did that inform you, or what did that lead to, as far as what was the next step here?
What was the next step? Yeah. So what we found was this relationship with specifically the social cognition subscale of the SRS, which makes a lot of sense given what we know about the role of the frontal cortex in cognitive functioning. And so the thing about the study that I just mentioned is that it was performed in two groups of largely male children. And so, as you know, there's a large male bias in the diagnoses of autism. And so, we sort of started with this group of mostly male individuals. But what we really wanted to do was to move on and also look at whether there might be sex differences in the effects of these oxytocin receptor gene SNPs on brain connectivity. And so in the next study that we did, we worked with the GENDAAR Consortium, which is a group of researchers at different universities across the US who worked to gather a large neuroimaging cohort of girls with autism and without autism. And so in our next study, what we did was a similar set of analyses, but this time looking at sex differences in the effects of the oxytocin receptor gene on reward network connectivity.
Yeah, this is wonderful data. What comes after this is just wonderful data, I think, in the role of autism. Before we get into the data, can you explain to us about GENDAR?
Sure, yeah. So this was a collaborative effort across a couple different research institutions in the US in order to recruit and collect data from a large cohort of autistic girls. And so the four different sites for GENDAR, and GENDAR stands for Gender Exploration of Neurogenetics and Development to Advance Autism Research, just so everyone knows what that stands for.
Thank you.
But so the girls were recruited and participated in MRI scanning at four different sites at Harvard, Seattle Children's Hospital, UCLA, and also Yale. And so the data came from these various sources and really gave us the opportunity to examine what was, when this data was published, the largest study of neurogenetics in girls with autism.
There's some powerhouse universities there too involved.
Yes, absolutely.
Let's talk about some of those scannings, or the scanning, and the connectivity with this data.
Sure, sounds great. So, similar to the previous study, everybody came in and had an MRI scan. These MRI scans are done, I should have mentioned, while everybody is at rest. And so people come in and have an MRI scan, but what they're actually doing is just looking at a little black sort of crosshair plus sign on a white screen. And they're told that they can just let their mind wander while we take pictures of their brain. And so we use those pictures, as I mentioned, in order to look at these co-fluctuating signals across different brain areas. And so here again, we're looking at co-fluctuating signals between the nucleus accumbens, that hub of the reward network, and other brain regions.
Okay. And some interesting findings, I think, is how the autistic girls, one, differs from the autistic boys, the similarities maybe between autistic girls and the typicals.
Right. So what we found was that, similar to what we had found just generally for connectivity in the male groups, that when we looked at what other brain regions the nucleus accumbens was connected to, was really robustly connected to other reward-related brain regions and also frontal cortex. And then again, we found those negative associations with the thalamus and visual areas. And that was both in autistic girls and non-autistic girls. So the next thing that we wanted to do was to look to see whether there may be any differences between those two groups, and we actually didn't find any differences, just looking at connectivity of the nucleus accumbens more generally. And so the next thing that we did was to look at how these oxytocin receptor gene variants might influence this connectivity. So what we found was that in the non-autistic group, having more autism-associated variants on the oxytocin receptor gene was associated with increased connectivity with frontal brain regions. While in the autistic female group, we actually found that more of these oxytocin receptor gene variants was associated with increased connectivity with other subcortical brain regions, including the caudate and the thalamus. And so, like we mentioned before, these are two brain regions that are very important in implicit learning. And so we wanted to see what this connectivity might be associated with. And again, we looked at associations with other behavioral measures. And what we found was that higher connectivity between the nucleus accumbens and these subcortical areas was associated with higher scores on the ADOS repetitive behavior scale, which we thought was quite interesting.
That is interesting. It kind of leads me to a question of, well, maybe one, what draws these individuals, these autistic girls, into an assessment, and what do their assessment results look like to receive that diagnosis?
Right, right. I mean, so overall, we did find that the autistic girls had higher SRS scores relative to the non-autistic group. But, as you know, there's sort of ongoing research in terms of the diagnostic criteria that goes into identifying girls and boys with autism. And so one thing that you may have talked about already is the female protective effect, or this hypothesis of a female protective effect in autism, which sort of suggests that it's possible that females may require a higher number of genetic variants or environmental factors to occur in order to sort of express the autism phenotype relative to males, so that females would require relatively more of these different factors in order to actually go ahead and get a diagnosis.
Okay. It appears their social motivation is much different than autistic boys, and pretty similar, if not equal to or greater than the non-autistics. And something else that I was wondering is if that social motivation is actually above the normal, where they might need more affinity or more social interaction, and if that is a result of the biology that gives them autism, or what leads them into the assessment.
Yeah, that's really interesting. I haven't done a lot of research myself on differences on the SRS or social motivation in girls with autism, but I think that's a really interesting point.
Yeah, okay. It's also the question about how girls camouflage or mask.
Right. Yeah, I think that leads in really well to actually the next finding. Maybe I should talk a little bit more about the
Yeah,
next finding in the
yeah.
paper. Okay, great. Um, so the next thing that we wanted to do was to compare autistic females and autistic males. So how might this, differences in the oxytocin receptor gene be differentially modulating brain connectivity in these two groups? And so what we found was that relative to autistic males, our autistic females showed upregulation of connectivity between the nucleus accumbens and the frontal brain region. And so this was really exciting to us because there was, really a lot of overlap between this frontal brain region that we found was upregulated in the autistic females and the brain region where we had previously found in the 2017 paper that higher connectivity in males was associated with lower SRS scores. So what we did again here was we took connectivity between the nucleus accumbens and this frontal brain region, and we plotted them as a function of the SRS. in our girls with autism. And so what we found was that really mirroring the results from 2017, again, having more connectivity between the nucleus accumbens and frontal brain regions was again associated with lower scores on the social responsiveness scale, sort of suggesting that there might be this sort of neurobiological compensatory mechanism that may facilitate some of this sort of masking behavior that you were just mentioning in girls with autism, given that we're finding this increased connectivity between frontal brain regions involved in cognition and cognitive control and these subcortical brain regions involved in reward and social processing.
Wow, that's quite the findings as far as recent autism research, and especially the gender differences. With those findings, how do you anticipate that informing the assessment side or um, the instruments used for diagnosing autism?
I mean, I think what it really indicates is that there are probably other, um, genetic and environmental factors that interact with the oxytocin receptor gene that cause these that are associated with these sex differences, right? And so, what those are, I think, is still an open question. And so, I'm not sure how this might impact use of the SRS in order to, and SRS is not a diagnostic instrument, in fact, for autism. The ADOS and the ADI are the gold standards for
Oh,
autism
definitely.
diagnosis. But what it sort of suggests is one biological mechanism that might be sort of facilitating enhanced social behavior in girls with autism.
This female protective effect, findings like that is explains a lot. It explains a lot about just the recency of autism, both the increase of assess or diagnoses, as we're down to like 1 in 36 or whatever, and
Mhm.
then just what we knew about that boy bias.
Right.
It's
Yeah. So there's been some previous research showing that, there are differences in gene expression in males and females. This may be one thing that also interacts with autism: genetic risk, differences in hormone levels, differences in, environmental inputs. And so there are a lot of factors, I think, that, need to be investigated in order to better understand some of these sex differences, and especially potential sex differences in the influence of autism-associated genetic variants on brain development. I do also want to mention that, in this study we were looking at one gene, right, the oxytocin receptor gene, but there are actually many genes, as I mentioned, across the genome that are associated with autism. And so more current research is taking more of a sort of genome-wide approach in order to look at genetic variants and how they might be related to brain development. And so, more recently, we've been doing things like looking at polygenic risk scores for autism, which really add up. So the idea there is that you would add up the sort of effects of different autism variants across the entire genome to
okay.
generate a single score. And then you could look at that and see how it's related to brain structure and function across development.
How easy is that to do? Is that something that's complicated per individual, or?
So I mean, there are a couple of steps. The first thing that you really need is a very well-powered genome-wide association study of autism. And so currently, our current GWAS of autism are still relatively limited in their power to detect the genetic variants associated with autism compared to other psychiatric diagnoses, for example, schizophrenia, where there are very large GWAS studies. And so, we really need to continue increasing our sample size in order to be able to detect all of these different genome-wide genetic variants that might be associated with autism. And then from there, it's sort of a matter of having having, as I mentioned, the well-powered GWAS, and then also having genetics data of your own in another cohort. So at UCLA, we often collect saliva in order to do genotyping from the participants that come in. And so when you have genotype data from your own individual group of participants, you can sort of marry those two different sources of data in order to make a score indicating, genetic predisposition to autism in the data that you have from your own unique participants.
Wow, that's amazing. That's great insight. I'm glad you shared that, because that's information that is hard to come by for the lay public. Just that information. Something with the oxytocin is very interesting, though, because of the everything it's attached to, both in the central and peripheral nervous
Mm-hmm.
system.
Mm-hmm.
So one, I guess it seems like that would be hard to narrow down, but two, the amount of roles it has seems like a great contender on that end.
Yes, it has a lot of roles. You're so right. So the oxytocin receptor gene, when it's bound to, sort of through a cascade of events, ends up impacting intracellular calcium levels. And so, that can promote the release of different neurotransmitters. Oxytocin receptors expressed on excitatory neurons can promote the release of glutamate, which leads to enhanced excitatory neurotransmission. For example, in the hippocampus, oxytocin has been found to enhance excitatory synaptic transmission and also help with learning and memory. Oxytocin receptors are also expressed on inhibitory interneurons, where they can influence the release of GABA, which is a main inhibitory neurotransmitter in the brain. And so that is thought to be involved with some of the sort of anti-anxiety effects that it has by working on inhibitory control in the amygdala. And then in the prefrontal cortex that we were talking about recently, oxytocin can influence both excitation and inhibition. So it definitely has many roles, and also, the roles are sort of dependent on brain region as well. So leads up lots of different potential avenues for investigation, I think.
Yeah, yeah. Oxytocin is a very fascinating molecule in our body. After this data, what kind of information could you share about how somebody in the lay public can find this information and kind of develop their understanding of the spectrum now that this word spectrum has kind of opened up?
Right. No, I mean, I think what you're doing here is fantastic. Bringing, scientists on to talk about their work. It's definitely a challenge. A lot of the work that we publish is in different scientific journals. It's not always accessible to a lot of people. I think scientists are also trying to do a better job of actually communicating with the public. Through social media and other means, right? I think also since COVID, we've had a huge opportunity to record lectures and talks and presentations at different universities and post them online. Going to the websites of different autism centers and research groups are all things you can do in order to learn more about this area of research. But I think what you're doing here is really fantastic. So happy
Oh,
to have
thank you.
been able
Yeah,
to.
it was one of the things that motivated me. It's having reviewing scientific literature and then bringing scientists on like yourself and
Mhm.
sharing such wonderful data. What are some of the things that you're currently working on?
Yeah, so a couple of things we're working on. One is, again, these large scale genome wide association studies of autism. And I am particularly passionate about performing that research in diverse populations. And so a lot of what we know about the genetics of autism and a lot of different psychiatric disorders right now is really focused on European populations. And we also know that what we find in these European populations might not always, sort of be portable to other populations. from sort of a precision medicine perspective. So your ability to predict using genetics is impacted based off of the different populations you might be looking at. And so what we really need is larger, more diverse genetic studies of autism, as I mentioned, in order to identify these different variants that might be associated not just with autism, but also with other, co-occurring issues. For example, sensory processing problems, motor related delays. And so there's a lot of room, I think, in order to improve in that area. So that's one of the areas that we're currently doing some research on. And then another area is, everything we talked about today is the impact of these common genetic variants, or SNPs, on brain connectivity. But what's the mechanism through which those SNPs are actually influencing brain development, right? So we know that genetic variation in these different SNPs can also influence gene expression. And so what we're also working to understand is how these different genetic variants might impact brain gene expression, and then how that gene expression in turn influences developmental trajectories during childhood and adolescence.
Wow. It seems like such a crucial time for that.
Yeah, it's really a great time to be doing this type of research, because a lot of these really large scale data sets have recently become, publicly available. None of this work could really be done without the participation of the community. Um, but yeah, it's a great time for doing this type of work. A lot of the different genetic variants that we've been talking about today, and the genetic variants identified through GWAS of autism, each individual variant has a very, very small impact, a very small effect size. So in order to identify these variants, you really need very well-powered, large-scale studies. And so I think we're sort of, we're in that era now where a lot of these studies are available, and I think it's going to be great for facilitating our understanding of autism and lots of other disorders as well.
Yeah, it's something that makes me anxious and excited about just seeing what happens next. Where do you think autism is in 10 years?
In 10 years, I hope we're working to, just understand the unique biology and needs of the individual, and that we're able to, where helpful, intervene earlier on before the sort of manifestation of really difficult and what can be, impairing symptoms, and that we would know earlier on in development who is more likely to go on to need, more targeted interventions for some of the different symptoms, and that our work in genetics and behavior and brain development can help to inform some of those findings.
Yeah, absolutely. That's where it's happening. I'm thinking about um, April 2024 Communications Biology from UCLA released a study on 6-week-old, and they followed them up to 12 months, and that was some revealing data about how the 12-month old they started developing autistic traits using an instrument. It wasn't an ADOS, I don't believe. I can't remember. But studies like that, and then the intranasal option of oxytocin seems like it has a lot of promise. It's very low risk, which is phenomenal. It's very inconsistent, but it does show that it does help some, and so that's a huge step.
I agree. Yeah, I think this work really helps to it just inform our understanding of how variation in human genetics can lead to variation in brain development and behavior. Everything that we talked about today, all of the individuals who participated were children and adolescents, and so I think you're sort of alluding to moving to younger ages is, very timely as well.
Yeah.
At this point, a lot of that data, the infant data, is still The sample sizes are relatively small, and so that's one area that I think we need to work on, increasing sample size there so that we have lots of power in order to identify different associations. But it's definitely a really interesting time to be working in this area. All the data from the different modalities seems to be coming together, the genetics, the neuroimaging, and the behavior. And I think that's really, really interesting and can be really informative for our understanding of heterogeneity in autism and other traits.
Yeah. I think that was very well said about different disciplines are coming together and working on kind of a common goal,
Mm-hmm.
and that's going to really just accelerate information. So that's very promising. Um, well, I can't thank you enough for coming on and sharing this wonderful data and being so kind, um, and just letting us know what to expect from you and the autism scientific community over the next few years. Everything looks promising, and maybe we'll just hold you up to that.
Yes, thank you so much for having me, and thank you for what you're doing here, spreading information and inviting me on. It's been a great experience, and I know a lot of people are learning from you and what you're doing here, and so appreciate you, and good luck moving forward.
Oh, thank you. Thank you so much. I hope you have a wonderful day and good luck with everything.
You too. Thank you.
If you are listening and enjoy the episode or enjoy the podcast, please feel free to leave a review or rating. In podcasting, reviews, ratings, and downloads are huge, and I very much appreciate your feedback. You can contact me at info.fromthespectrum@gmail.com. And thank you for listening to From the Spectrum podcast.